Hello, virologists, and welcome to Virology Online.
I'm Teiana. I'm Colette.
I'm Mira.
And I'm Claire. We are your hosts today, no pun intended, and we're going to be talking about the usage of an adeno-associated virus as a vector in an exciting new form of gene therapy that treats adults with hemophilia B, a bleeding disorder.
So what is nanotechnology? Nanotechnology can be defined as the use of nanoparticles such as atomic, molecular, and supramolecular particles for industrial purposes. To make this more applicable to virology, viral nanotechnology is when a virus is used as a nanoparticle for biomedical purposes. Viruses have been studied as deadly pathogens, which cause disease in living forms, but through manipulation in the 1970s, scientists discovered a way to use viruses as a tool to our advantage, such as their role in expression systems in biotechnology and in the fundamental cellular process. Viruses were finally able to be used as a vector for the benefits of human through nanotechnology in things such as gene therapy, cancer control, and to control damage of organisms in medicine and agriculture. Nanotechnology is important not only for the reasons just stated, but because it allows imaging at the subatomic level that scientists and researchers never were able to have before.
One way researchers have been attempting to treat diseases has been gene therapy, which is unique in that it targets the direct causes of genetically based diseases rather than merely treating the symptoms. It can attempt to activate gene expression for diseases caused by a lack of expression of certain genes with essential protein products, or it can silence genes that have harmful effects. This is an incredible form of biotechnology that has the potential to treat genetic diseases at their source, the genome. Researchers can also use nanopore DNA sequencing, in which nucleic acids pass through a nanopore that tracks changes in electrical currents, and analysis of these changes can be used to sequence the DNA in real time. Using nanopore sequencing provides researchers the opportunity to analyze integration sites of viral genomes and see the effects of using viral genomes in human DNA, which we will cover more in later slides. The disease we're going to be looking at today is called Hemophilia B, and you may have heard about it if you've taken a genetics class because it's a classic example of a sex-linked recessive trait, or maybe if you've taken a European history class due to its prevalence amongst royal families associated with interfamily marriage and thus inbreeding. It's monogenic, meaning the disorder is caused by mutations or damage to a single gene, and that gene is known as F9. It's associated with the production of FIX, or coagulation factor 9. When mutations occur on this gene, it causes a deficiency in FIX, a blood protein which acts as a coagulation factor to decrease bleeding and promote wound healing. Without sufficient FIX proteins circulating, those with hemophilia B experience excessive bleeding and hemorrhaging, which can lead to permanent joint damage. Because it's X-linked, it can only be passed from a carrier mother to son through the X chromosome, but the altered gene can occur spontaneously without family history in about 30% of cases. Typically, hemophilia B is treated with injection of recombinant FIX or with infusions of human blood and/or plasma, but as we will continue to explore throughout this video, scientists are looking into ways to treat it with gene therapy instead.
The study we'll be looking into today is called Gene Therapy with Adeno-Associated Viral Vector 5 Human Factor 9 in Adults with Hemophilia B. In this study, 10 adults with hemophilia B and the severe bleeding phenotype were chosen to participate. These 10 subjects were split into two groups, the lower dose cohort and the higher dose cohort, with each group consisting of 5 subjects. The cohorts were given different concentrations of a single dose of AMT-060. So what is AMT-060? This is a combination of adeno-associated virus 5 and a liver-specific protein that drives the expression of a factor 9 gene, which, as you'll remember, produces the FIX protein. The purpose of the study is to evaluate the amount of FIX activity in the lower and higher dose cohorts, using viral nanotechnology to manipulate the genome of the subjects. This study is evaluating whether this will be a safe and effective treatment for people that have hemophilia B and have severe bleeding phenotypes.
So what are adeno-associated viruses and how are they used in the context of this study? Adeno-associated viruses are small viruses that typically infect humans and other animals but aren't pathogenic. They are called adeno-associated because they require adenoviruses to become pathogenically activated due to factors from the adenoviruses. The adeno-associated virus is commonly used in genetic therapy due to its low side effects for the patients, only causing mild immune system response, and are also used because they are site-specific and integrate into the host genome at specific points, allowing researchers to control where they enter and then track their activity.
In this study, AMT-60 technologies consist of an adeno-associated virus vector 5. The AAV5 vector was manufactured in insect cells using baculovirus expression systems. In this vector's genome is a small gene operon which includes the wild-type FIX gene under the control of LP1, which is a liver-specific promoter. This drives production and replication of the viral genome in liver cells, where FIX is typically produced. This vector has been shown to be effective in previous gene therapy treatments for gene addition uses and doesn't elicit a strong cellular immune response against the capsid. In this study, the hypothesis that these scientists had was that the liver tissue-specific infection and gene transfer mechanism performed by AMT60 offers a constant and sustained endogenous reproduction of functional FIX, which should reduce FIX activity. In their experimental design, they administered AMT60 in a 250 milliliter peripheral intravenous infusion for 30 minutes. Patients were treated in 2 consecutive cohorts. Cohort 1 received a 5 × 10 to the 12th genome copies per kilogram, which was considered the low dose, and cohort 2 received 2 × 10 to the 13th genome copies per kilogram, which was the high dose. The vector titer to make these doses for each cohort was done by using qPCR to determine the genome copies per kilogram that each patient received. Participants were monitored after the first 24 hours after administration, and the FIX prophylaxis proceeded for 6 to 12 weeks after administration. And FIX activity from patients were measured before and after the AMT treatments were administered.
The results of this study show that all the patients showed an increase in FIX activity, which helps blood clots form to prevent excessive bleeding. The lower dose group showed an 81% decrease in FIX medications and shows a decrease in spontaneous bleeding rate by 53%. On average, the patients in the lower dose could now be classified as having only mild hemophilia. The higher dose was given to the group with the more severe hemophilia pre-treatment. The higher dose group showed a 73% decrease in FIX medications usage and a 70% decrease in spontaneous bleeding rates. On average, the higher dose could then be classified as having moderate hemophilia. Both groups showed significant improvement, supporting the hypothesis that AAV5 is a successful treatment for hemophilia.
Finally, here is a list of our sources, including our primary research article listed second. We hope you all learned something about the use of viral nanotechnology in gene therapy. Thanks for listening and have a great day.